to marine life represents a new source of opportunity to expand medical and scientific knowledge.
We're changing the trajectory of the community, both through bringing science to the community, making new discoveries that will have an impact on fisheries, on human health, and also changing the trajectory of the students we train at the Academy.
Coming out of the Academy, I have more hands-on experience than a lot of my colleagues that did 4 years in college. It's a small commitment that unlocks life-changing opportunities.
What's really exciting about GMGI is the way the technology has progressed. We're on the cutting edge, and that will allow us to answer these really complex and interesting questions.
We can find new discoveries that can directly impact human health.
We are small but mighty. You would never know that we are a team of under 30 people when you see the groundbreaking research being conducted in our labs and the transformative education experiences being provided at our academy. And we are just getting started.
We really have a true gem in GMGI. It's not, as you could see, it's this two wings to the program, this just astonishing educational program. I've taught for 47 years and done a lot of work and had a Howard Hughes professorship to try and develop something. So I've done a bit of stuff, and I've talked about this program at places like Howard Hughes Medical Institute and around the country when I'm going places, because I think it's so amazing. Taking these students who you could see often just high school education, and that's about it, or maybe a year of college, didn't seem to know where they were going, and turned them into these students with a future. They're enormously skilled as biotech technicians. They're fueling this biotech industry around here, which hopefully we're going to be able to grow around Gloucester. But John Doyle and his team they've done more than that, and it's, teaching is about mentoring, and if you talk to these students, you get a flavor of it there, but they found their confidence, they get to feel empowered, and so many of them have just taken off. They've climbing the ladders within these biotech companies that they've gone to. A number have decided to go on to grad school and just get heavily into science. It's been probably the most transformative educational thing I've seen, and that's half of what GMGI does. I'm here to talk about, introduce Andrea Bodnar tonight, who's going to give you a sample of the other part of what goes on at GMGI. This is an utterly world-class institute. Dave Walt and Mark Vidal, who you saw in there, are hugely important scientists. Dave Walt's not a professor at Harvard, but he founded the company Illumina, which is sort of the big pusher of what you've heard of as next-gen sequencing, which just drives so much of biological research. Mark's an incredibly skilled cell biologist at Harvard. And the commitment from those two is wonderful, and you see it reflected. The company. Those of you who heard Tim O'Donnell talk in the first one, that they call it a metagenomics approach, where you take a sample of seawater or a sample of something, sequence all the DNA from all the organisms, and then work back computationally and comparing sequences to find out what was there. I was just at the meeting of the National Academy of Sciences on the weekend, and they had sort of trying to pick out futuristic projects, and people use exactly the same thing they're using here to work out the skin biome, the pathogens and commensals that we have on our skin, to work on ecosystems to find out about climate change, or some of you may have heard about what Greenland was like 2 million years ago in the news reports. They drilled 3 kilometers through the ice, took a sample of stuff down at the bottom, and did exactly the kind of thing Tim and GMGI are doing to work out everything that was down there, including some animals that are no longer with us anymore. So it's really cutting edge stuff. Andrea Bodnar is the Donald G. Combe Science Director, and she oversees this whole scientific part of the program. There are 3 major foci. One's biomedicine and biotechnology, and you can see why that's here. And there's a lot of interest in growing biotech around here, provide a new source of good jobs for our local folks when they can't go fishing anymore. functions on ecosystems and on human health, and we'll be hearing some of that tonight. And then, as you heard from Tim, new ways of doing fisheries management. I'm sure the techniques you've heard him do are going to be all around the world before you know it. So Andrea, this is a special treat for me. He's one of my special friends, as well as a scientist I admire, is a trained biochemist, and her interest now at the interest of sort of intersection of marine biology and health. And she had quite a journey getting here. We both went to undergrads universities in Ottawa. I was at Carleton University. In Canada, I was at Carleton University, Ottawa, and Andrea went to McMaster University in Hamilton, where she did her PhD. And then her travel started. She went from there to do a postdoc at the University of London in the Department of Neurological Sciences, and then kept progressing. She went. She was in the oncology department at Hoffmann La Roche, the big pharmaceutical company, the Department of Cell Biology at the Geron Corporation, which specializes in blood cancers, and then the Bioprocessing Technology Institute at the University of Singapore, where she was doing sort of high-tech stuff. But her husband, John, does finance, and then that led her to Bermuda because of him being there. And she had to find something to do, and there was the Bermuda Institute of Oceanographic Science, and she discovered sea urchins, which we're going to hear about tonight. And it was actually special for me because I'd organized a big DNA repair meeting there once, and I went to visit the place, and I ended up meeting Andrea, and we became friends. And so I have sorry, this is, we have to probably touch this slide to get it activated. Okay. Yeah, so I'll show you this slide again. Andrea was invited, before she became director, to a symposium in 2015 out near Lanesville. And you could tell we were friends. We were already standing beside each other by then. So I think they made an incredibly astute choice bringing here. And I had found a picture when we were all visiting there back in, before she came here. So anyway, Andrea's done an amazing job here. She's overseen this multidisciplinary, multi-technique sort of collection of science. She's written the key papers on the lobster genome, on a test for the white-spotted syndrome shrimp virus, on they sampled everything that's down in the sediment in Stellwagen Bank. It's an amazing breadth that she's a senior author on all of those, in addition to the work she's going to tell us about. She's an amazing mentor, and she's just a terrific science. And just before she starts, I'll say one thing to those of you who don't do science all the time. You know, most would all love to learn about humans, but they're kind of tricky scientific subjects. Geneticists can't get them to mate with the person you'd like. You know, even your parents can't make you do that. So you can't edit people unless you're trying to solve a disease or something. So scientists have used what they call model systems and that other organisms, and that makes sense because life started about 3.8 billion years ago, and it didn't really diversify significantly until about 1 billion years ago. So we're so much alike down at the molecular level, and you can sort of choose your organism for the thing you want. And I think Andrea's made an incredibly astute choice as to if you want to study aging, you want to hear about this research. So hold your questions afterwards. You won't need me anymore, because if you've watched Andrea at any of the science hours, she's wonderful at leading a discussion. And Andrea, come on up. She's going to she'll tell you about her beloved sea urchins, and I think let you in on their spiny secrets of longevity, right?
Wonderful. Thank you so much, Graham, for the very kind introduction, and thank you, everyone, for joining us this evening. As you just heard, I have a very unconventional scientific career, but I've always been interested in questions related to human aging and cancer cell biology. So having ended up at a marine biology institute in 2003, I immediately became captivated by the long-lived animals that live in the ocean, and wanted to understand how these animals can live for centuries in the absence of any signs of aging. And for many of these animals, there's no or very few reported cases of cancer. So I really wanted to understand not only how these animals live for so long, but more importantly, I wanted to understand how these animals remain healthy throughout their long lives. And I decided to use sea urchins as a model to try and answer some of these questions. And ultimately, what I hope to do is to learn from nature of how she solved this problem of aging and apply something of what we learn to preventative or therapeutic strategies for human age-related diseases and cancer treatment. And so I'm going to spend the first part of my talk sort of informing you on why I chose sea urchins as a model, but I'm going to start off by trying to convince you that this is not an entirely crazy idea, and that marine animals have actually provided tremendous models historically for scientific research. They've really allowed us to answer some fundamental biological questions, and many of these are relevant to human health. So one way to illustrate this is to introduce you to some of the marine animals that have been recognized at the highest level in science with the awarding of Nobel Prizes. And so this goes back more than 100 years to work done by Eli Metchnikoff, who used sea stars as a model, which really formed the foundation of our understanding of cellular immunology and how our bodies defend themselves against bacteria and viruses. And this continues on with work with the squid as a model for helping us to understand how nerves function and how signals are sent along neurons in our nervous system. Next, we have the horseshoe crab, which was a fantastic model for understanding how our eyes function and how photoreception works. Next, we have the sea hare, which I think is one of the cutest little animals in the ocean, but it's also very smart and can exhibit the three basic forms of learning: sensitization, habituation, and classical conditioning. And it does this all with a very simple brain. And it's by coupling the ability to learn with a simple and well-defined nervous system allowed Eric Kandel to map all the neural circuitry associated with learning and memory using this animal as a model. The sea urchin has been recognized for two contributions to Nobel Prize winning research: Otto Warburg's work in the 1930s, which helped us to understand how cells make energy through respiratory chain function in the mitochondria, and also Tim Hunt's work, which discovered cyclins, which control cell division. Of course, we have to understand what controls cell division if we want to understand what goes wrong when we lose control of that process in the progression of cancer. And then last but not least, we have the jellyfish. And you can see it glowing beautifully green here. And the discovery of green fluorescent protein has really revolutionized the way that we can visualize research and medicine processes that are going on. And so all of these animals had particular adaptations that allowed these great discoveries to be made, whether that was transparent embryos or large nerves or large rods and cones in the eye, synchronized cell division, or the ability to learn. But the adaptations that I'm really interested in are life history adaptations. How long things live, when they reproduce, whether or not they age, and their resistance to disease. And when you look across the animal kingdom, which is what's really striking, is there's a tremendous range of natural lifespans, ranging from just minutes to millennia. And so on the short end of the life history spectrum, we have this creature, the mayfly. It's the shortest lived animal that we know. There are about 1,000 species of mayflies, and they all live less than 24 hours. But this one is the short-lived record holder, Delania americana. The adult female lives for about 5 minutes. She hatches, mates, lays eggs, and dies in 5 minutes or less. At the other end of this spectrum, we have this beautiful little jellyfish, which is reported to be the only immortal animal on this planet. So this animal has an incredible life history, and it can go from this fully formed adult jellyfish. When conditions are stressful, it can reverse development and revert back to an embryo and wait things out till things improve, and then develop back into an adult, and then revert back to an embryo, and then back to an adult, and then back to an embryo, continuously into perpetuity, and therefore it has achieved immortality until, of course, something eats it, which only goes to show you that immortality is pretty much impossible to achieve. But then nature has really provided us with pretty much everything in between these two extremes of the spectra. Now, most people who study aging focus their attention on the short-lived animals, the worms, the mice, and the flies. And this is a very sensible choice. You can do some experimental manipulation, and within the scale of a grant, you can tell whether that animal lives longer or shorter. But I've become fascinated by the animals at the other end of the spectra. And as you'll notice, all of these animals are ocean-dwelling animals. In fact, all of the Earth's longest living animals live in the ocean. And this goes for the longest lived mammal, which is the bowhead whale, reported to live more than 200 years. The longest lived vertebrate on the planet is the Greenland shark, which can attain a lifespan of nearly 400 years. And there are many long-lived invertebrates in the ocean. One of the record holders is the ocean quahog clam, with the oldest individual so far identified being 507 years old. But there are many, many other long-lived animals in the ocean. Very familiar to us in Cape Ann is the American lobster, reported to potentially live for 100 years. The geoduck clam, the oldest individual so far identified, has been 168 years old. The red sea urchin makes this list because it can live for over 200 years, and the rough-eyed rockfish is a deep-dwelling fish that can live for more than 200 years. Now, these are not the longest-lived animals in the ocean. The ocean Methuselahs actually go to the sponges and the corals, and of course the immortal jellyfish. These are animals that can live for more than 1,000 years. So a number of species of sponge have been recorded to live for 1,000 years. The great barrel sponge is one of those that can live for more than 2,000 years. There are several species of corals that can live for thousands of years. The deep sea black coral has been reported to live more than 4,000 years. So these animals are remarkable, and I'm sure there's something really interesting we can learn from them, but I'm not focusing my attention on them only because they're very distantly related to humans, and I'm hoping that something that we discover from these long-lived animals will translate to helping human biology. And so I'm going to stick to my original list with animals that are much more closely related to us. However, you'll see from these animals, I think it's pretty obvious that most of these animals present some pretty significant challenges to use as laboratory models. I mean, I'm really not going to keep a colony of Greenland shark in the lab to study. But there's an exception here, the red sea urchin. Sea urchins have been a model for scientific research for more than a century. They're easy to maintain in the lab. There are hundreds of labs around the world who use these as a model. We know a lot about their biology. I think they present a really unique opportunity to start to understand the cellular and molecular mechanisms that are underlying extreme longevity and healthy aging. Sea urchins are a classic model of developmental biology. It's very easy to get egg and sperm from the sea urchin, and if you mix those in a beaker of seawater, baby sea urchins form within a couple days, going through all of embryology, from a fertilized egg to a fully formed larvae, which has a functioning digestive system, nervous system, skeleton, its immune system, and that all happens in a couple days. So a lot about what we understand about fertilization and early embryonic development comes from studying this animal. And one of the reasons they've been a great model is, genetically speaking, they're very closely related to us, which I think is one of the most humbling lessons I've learned since switching from human biology to studying sea creatures. So this is just a tree of life that I've taken from the National Geographic website. And basically the branches of the tree are just showing how related animals are to one another. And this tree of life is showing the two branches of the animal kingdom. The green branch over here are the protostomes, which include insects and worms and mollusks. The brown branch over here are the deuterostomes. And at the top of this tree are humans and other mammals and vertebrates. But you'll notice that just below this branch are sea urchins and other echinoderms like sea star and sea cucumber. So we're very closely related, which means that discoveries made in these animals may more easily translate to humans than discoveries made with animals that are more distantly related. And sea urchins are commercially fished. And so from commercial fisheries, we know a lot about their life histories, their reproductive patterns, and their susceptibility to disease. And it's when you look into the fisheries data that things become very interesting from an aging perspective, because different species of sea urchins have very different natural lifespans. There are some species of sea urchin, like the white sea urchin, which is reported to live for only 2 years in the wild, where other species, such as the red sea urchin, can live for more than 200 years in the wild. And then we have pretty much everything in between. And so really we can use a comparative approach to try and understand why this animal is only living for a couple years, where this closely related cousin is attaining a lifespan of more than 200 years. And not only is their range of lifespans remarkable, but the other really interesting thing about sea urchin biology is they don't appear to age. They exhibit what we call negligible aging. And what I mean by that is these animals grow and reproduce throughout their lives. There's no obvious physiological decline associated with getting older. There's no increase in mortality rate associated with getting older, and there's no increased incidence of disease associated with getting older. And that includes no reported cases of cancer in this animal. And so for all of these reasons, they really present a great model to try and understand the mechanisms underlying healthy aging and resistance to cancer. Now, of course, this is a very, very different life history from humans and other aging animals, where we go through a very characteristic physiological decline, increased probability of death, increased incidence of disease as we age. So people studying humans and other animals that age have come up with 9 hallmarks of aging. And these are done not only studying humans, but also studying a lot of short-lived animals that age very quickly, like this worm, C. elegans, which lives for about 3 weeks, the fruit fly, which lives for about 3 months, and the mouse, which lives for about 3 years. And by studying all of these aging animals, they've come up with these hallmarks of aging, which we've been using as a framework to try and understand how sea urchins differ from aging animals. So there's a lot of things going on here, so I'm just going to focus on 3 of these hallmarks of aging, and I'm going to very briefly review what we know about sea urchins compared to humans, because I really want to focus the last part of my talk on some of our new data on the genetics of aging. So one of the hallmarks of aging is telomere attrition. So telomeres are the repetitive sequences of DNA that are found at the end of all of our chromosomes, and they play a really important role in protecting the integrity of our genome. So when I speak to a lay audience, I usually compare telomeres to those little plastic tabs that are found at the end of your shoelace that prevent your shoelace from unraveling. Telomeres do the same thing for our genome. They provide integrity to the genome. The problem is every time a cell divides, we lose a little bit of telomeric DNA from the end of our chromosomes. So as our cells divide, as we grow older, as we age, our telomeres are getting shorter and shorter, our chromosomes are getting shorter and shorter. When telomeres get critically short, a message is sent to the cell to say, that's it, I've run out of telomeres, I'm permanently going to stop dividing. And it's thought to be the accumulation of these cells with short telomeres that can no longer divide that contributes to the aging process. So when we look at telomere biology in sea urchins, we find a very different story. They don't shorten their telomeres as they get older. They express an enzyme that allows them to rebuild their telomeres back after every cell division. So they've solved the telomere problem of aging by maintaining their chromosome integrity as they grow. Another hallmark of aging is mitochondrial dysfunction. Mitochondria is the powerhouse of the cell, and it produces energy using oxygen as a terminal electron acceptor. As part of this process, there's a byproduct, which is the creation of something called reactive oxygen species. These are highly reactive molecules that damage all the bits of our cells, DNA, proteins, and lipids. And over time, although our cells have some repair capacity, our repair capacity is not 100%. So over time, we accumulate oxidative damage in our tissues, and that contributes to the aging process. When we look in sea urchin tissues and try and measure oxidative damage to their proteins, lipids, or DNA, we find 2 really interesting things. One, they have very, very low levels of oxidative damage, and that level does not increase as they age. So once again, they've solved one of the hallmarks of aging. And the third thing I want to point out here is stem cell exhaustion. So stem cells are these remarkable cells that we have that allow us to replenish tissues. And there are some tissues in the human body that are continually replenished throughout our life. So for example, our blood and our skin are always turning over. And that's fueled by stem cells that are replacing cells that we lose. However, as we age, we lose both function and number of stem cells, so our ability to regenerate tissues declines as we age. So when we looked at tissue regeneration in sea urchins, again, we see something remarkable. Sea urchins and other echinoderms, so related animals, have a tremendous ability to regenerate tissues. And we can cut off the external appendages of the sea urchin, its spines and its 2 feet, which are its sensory and motor appendages, and sea urchins will continually grow those back again. And old animals will grow those back as efficiently as young animals, and long-lived species will grow those back as efficiently as short-lived species. So once again, they've solved one of the hallmarks of aging. Now, the problem with this approach is that it's not teaching us anything new, except to show that sea urchins don't age like other animals. And the other thing is, it's not really helping us to understand why some sea urchins only live a couple years and others live for centuries, because both short and long-lived animals appear to have the similar biology. And so we decided to step back and take a more unbiased approach and just have a look at what the genes and the genomes of these animals can tell us. So for those of you who don't think about genetics every day, your DNA or your genetics is really providing the blueprint for all of the functionality of your cells in any organism. And the DNA is packaged inside the nucleus of the cell in chromosomes. And along the chromosomes, we have genes, which are functional units encoding proteins that have a particular role in the cell. And so there are a couple of things that we wanted to look at in sea urchins. We wanted to compare the genomes of short-lived species to long-lived species to see if we could see any differences that would help to understand why they have these different lifespans. And we also wanted to look at how they're using their genes as they age. So if they're not going through all these hallmarks of aging, what changes are occurring in these animals as they age? And we can do this by looking at gene expression. So both sea urchins and humans have about the same number of genes in our genome, about 20,000 genes, but we're not using all these genes at the same time in all of our cells. And that's what makes our cells different. That's why a brain cell has a different function from a kidney cell and a muscle cell, because they're only expressing a subset of genes to let them do their specialized function. And so we can look at what genes are turned on or being expressed as an indicator of the functionality of the cell. And so we've switched our attention to comparative genomics, age-related gene expression, and also doing functional genomics to actually test the function of genes. And so the first thing we wanted to look at was age-related gene expression. How does a pattern of gene expression change with age in sea urchins? So when I talk about gene expression, when you have a gene and it's switched on, it makes a copy of itself called mRNA. That mRNA then leaves the nucleus, goes into the cytoplasm, is converted to a protein, which then takes up its active conformation and does its job in the cell. So when we want to know what genes are switched on, what we do is we measure this population of messenger RNA or mRNA, and that tells us what genes are present. And when we look at age-related gene expression in aging animals, like the short-lived worm, fly, and mice, as well as aging humans, of course we see very different changes or profiles of gene expression from animal to animal and in different tissues, but there are some central themes that emerge from these kind of data. As we age, we see a decrease in expression of genes involved in energy production. We see a decrease in expression of genes involved in maintenance and repair functions, and we see an increase in expression of genes involved in stress response and inflammation. And this is sort of a profile, typical profile of an aging animal. So we wanted to see what's happening in sea urchins and how does their gene expression profile change with age. So we decided to focus on the long-lived red sea urchin and look at age-related gene expression. So we collected a number of young animals, 18 of them with an average age of about 4 years, and we collected some old animals, 16 old animals with an average age of about 130 years. We took some tissues from them, muscle tissue from the jaw structure, which is called Aristotle's lantern, esophagus tissue, and radial nerve cord. And we asked the question, what mRNAs are being expressed in the old tissues versus the young tissue by sequencing the mRNA populations. Now, when you do this kind of experiment with other long-lived animals, and this gorgeous creature is the naked mole rat. The naked mole rat is the longest-lived rodent. It lives for about 30 years compared to a mouse, which lives for about 3 years. And when you look at age-related gene expression in the tissues of this animal, they don't change very much over the course of its lifespan. So it's very stable gene expression. And that's what we expected to see in the red sea urchin as well. And indeed, For muscle and esophagus tissue, that's exactly what we see. Very few genes changed over the course of more than 125 years in this animal. Less than 150 genes were either up or down regulated with age, and that's out of the 22,000 genes that are in the genome. And we're looking at a change of about twofold, either up or down regulation, but very, very few changes, similar to what you see in the naked mole rat. However, when we looked at radial nerve tissue, we saw a very different picture. More than 3,000 genes are changing, either up or down regulated with age. Now, this was very unexpected and very curious and merited a little bit of a deeper dive. And so we had to look a little bit more carefully at what was going on in the nervous system of this animal. Now, the nervous system is pretty important. It's a central control system of any animal, allows us to sense the environment, respond to the environment, controls our thought, our motion, whether voluntary or involuntary, really controlling the whole organism. And the way it does this is through its nerve structure, where nerves are stimulated. And that sends a signal which is transmitted along this long axon until it encounters another nerve, where that signal is then transmitted to the next nerves through something called a synapse, through the release of neurotransmitters, which are then allow the signal to transmit through the next neuron in the system. Now, we know that our nervous systems decline as we age. We know that there's a decrease in expression of genes at the synapse. We know there's a reduced amount of neurotransmitter production. We know that there's an increase in accumulation of aggregates of protein in our nervous system. And I think most people are familiar with that in terms of amyloid plaques that accumulate in Alzheimer's disease. Now, when we look at the age-related gene expression pattern in the nervous system of the red sea urchin, it's quite remarkable what we see. It's almost the opposite expression pattern to what we see in aging animals. In the red sea urchin nervous system, we see an increase in expression of genes involved in synaptogenesis and maintenance of the synapse and neuroprotection, an increase in expression of genes that are involved in neurotransmitter production and transport, an increase in expression of genes involved in protein recycling. Autophagy and proteasome activity allow cells to get rid of aggregates of proteins and get rid of misfolded proteins, and those processes are upregulated in the nervous system of this animal. We see an upregulation in genes involved in nutrient sensing and regulation of metabolism, which is critical for aging, and also an increase in transcription factor and chromatin regulators, suggesting that there might be a gene expression program going on to preserve the nervous system of this animal. And so our conclusion from these studies is really that preservation of the nervous system might be key for longevity and negligible aging in these animals. Now, this makes a lot of sense when you think about it, because really, if you can preserve the nervous system, it's controlling the activities of many other tissues. And so really, the reason we probably see very little effect in other tissues is because the nervous system is able to maintain the balance. And if you look at short-lived model organisms, and you can improve this nervous system function in those short-lived models. For example, if you increase autophagy in the brain of a Drosophila, of a fruit fly, that animal will not only live longer, but it remains healthier. So just by altering nervous system function, you can live a long and healthy life. So that might be the strategy that red sea urchins are using. So we wanted to look again a little bit more at what might be causing the differences in lifespan between these animals through a comparative genomics approach, by comparing the genomes of these animals to say what's different about them. Now, at the outset of this, we had genomes for 3 species of sea urchins to compare. 2 short-lived species, Lidocynus variegatus, which lives for about 4 years, Lidocynus pictus, which lives for about 7 years, and the purple sea urchin, Strongylocentrotus purpuratus, which lives for about 75 years. But when we started these studies, we didn't have a genome for the red sea urchin, this animal that lives for 200 years. So that's the first thing we did at GMGI, was we took 1 male animal, we extracted DNA from testes tissue, and we were able to assemble that genome into its 21 chromosomes. And on these 21 chromosomes, we could predict more than 22,000 genes in this animal. So this now gave us these 4 genomes to start comparing to each other. And the first thing we noticed was that even though these animals have the same amount of genetic information, the same amount of DNA, they've arranged their DNA quite differently. So long-lived species have arranged their DNA in 21 chromosomes, and short-lived species have arranged their DNA in 19 chromosomes. And this is because when you look at the chromosome number 1 in the short-lived species, it's a fusion of chromosome 3 and chromosome 15 from the long-lived species. And chromosome 2 in the short-lived species is a fusion of chromosome number 7 and 11 from the short-lived species. And so they have this rearrangement of their genome. And also when you look at individual chromosomes, we also see that many genes, the order of many genes are rearranged in the genome. And so they have very dynamic differences between short-lived and long-lived species. And these are exactly the kind of differences that you would predict would lead to very different characteristics. However, we can't directly link that to lifespan. However, what these types of rearrangements typically can do is they can cause an expansion or contraction of gene families. And that's something that we can look at in these animals. And when we do that, we see some really interesting trends in the long-lived red sea urchin when we look at the expanded gene families. Most of those expanded gene families have a role in immunity. Many of those expanded gene families have a role in nervous system function. And the third biggest category is related to nucleic acid metabolism. So in the immunity category, there's an upregulation or an increase in the number of genes involved in pathogen recognition and also antiviral immunity, as well as regulation of inflammation, elimination of damaged cell and protein aggregates. And so having a robust immune system is a great thing to have. If you can defend yourself against infectious disease, it promotes survival, it promotes longevity. So this might be one of the keys to why these animals are living longer than the short-lived species. We also see an expansion of genes involved in the sensory nervous system. And this involves chemosensation, photorecept mechanosensation, and thermosensory networks. And really, again, if you're an animal who is really adept at sensing and responding to changes in your environment, that will promote survival and that promotes longevity. We also see an expansion of genes involved in maintenance of the nervous system. So again, maintaining your nervous system will allow you to live a long and healthy life. In the nucleic acid metabolism category, we see an expansion or duplication of a number of tumor suppressive genes and key genes involved in DNA repair. So these animals, these long-lived animals, are able to maintain integrity of their genome, and potentially this is how they protect themselves from cancer. Tumor suppressor genes are genes that control regulation of cell division, and what we see is mutations in these genes lead to cancer formation in humans. So having multiple copies of these, having multiple ways to keep the brakes on cell division is a great way to prevent cancer formation. And so really this combination of expanded genes would promote survival, longevity, and cancer resistance. And not only can we look at these sort of large-scale genetic rearrangements, we can actually ask what are the differences at the level of sequence or nucleic acids in the genome, so individual building blocks of the genome. And we do this by doing an analysis called positive selection. So this is when there's a change in the sequence of the genome that leads to a change in the protein, which is selected for in the long-lived animals. And when we do this, we see there are about 448 genes under positive selection. And the first thing we did was to ask, do any of these genes, are they known to play a role in longevity in other animals? And so we compared them to the list of longevity genes that have been identified in worms, flies, and mice and humans. And we see that there are about 16 genes that overlap in our data sets. And these are really key genes that play a role in insulin signaling, in nutrient signaling, in oxidative stress. And so it's really interesting that there is an intersection, and some of the methods that we know that animals use to achieve longevity are used by the red sea urchin as well. But this is only 16 out of 448 genes, so there's a lot we don't know about the genes in this animal that may be promoting longevity. And so we can ask the question, do any of these positively selected genes have any interaction with each other? And so that's where things become quite interesting because they actually fall into this beautiful network. So this is about 185 of those genes, which actually have some connection to one another. So here, each of the circles represents a gene product, and the branches or lines between them show that they interact with each other either physically, or they're co-expressed, or they have some connection biologically together. And so why this is interesting is it actually suggests that there may be a network of anti-aging, an anti-aging network. And why this is interesting is when you're thinking about a therapeutic approach, you certainly don't want to have to tweak the individual 448 genes individually to try and have a positive outcome. But if you have a network that you can look at and identify nodes in a network, if you can modulate activity at those nodes, it can have an effect on many gene products. Now, this network is really only makes sense if those genes are expressed in the same place at the same time, and we can look at gene expression for these genes, and indeed we find something very interesting here. So this is a heat map of gene expression in muscle, esophagus, and radial nerve tissue of young animals and old animals. Every line is a gene, and the color indicates how much gene expression there is. So if it's blue, that means it's low level of gene expression. If it's red, there's a high level of gene expression. And in muscle and esophagus tissue, we don't really see a pattern, but if you look at the last two columns, in radial nerve, we see a striking pattern. Many of these genes under positive selection are highly expressed in the old radial nerve tissue. And so they are expressed in the same tissue at the same time, allowing them to interact together as a network. So what are the secrets of longevity? Maintain chromosome structure by maintaining your telomeres. Minimize oxidative damage. Maintain the ability to regenerate your tissues. Protect your nervous system and be able to eliminate aggregates of proteins and amyloid in particular. An expansion of the sensory nervous system is probably key. Also an expansion of tumor suppressor genes and expansion of certain immune genes as well. So this, I think, is a recipe for a long and healthy life. However, we really now want to go in and test the function of some of the genes that have surfaced from these studies. So we have to take these from genomics to functional genomics. And we're doing this through a grant from the National Sciences Foundation to start to build the tools to do functional genomic studies in sea urchins. This is a collaborative grant with Gary Wessel's lab at Brown University and Steve Watt's lab at the University of Alabama, where we're starting to be able to modulate the gene function in sea urchins and look at the effect of that gene. And Gary's lab at Brown University is one of the first to use CRISPR technology to start to modulate gene function in sea urchins. In this experiment, he's knocked out a gene called PKS, which is responsible for the pigment formation in this animal, and you see that this gives rise to a beautiful albino animal. Steve's role in here is to improve the husbandry of this animal so we can do transgenerational genetic modifications. And the role at GMGI is to develop cell culture tools so we can look at gene function in cells in the lab outside of the whole animal. And so we're now beginning to develop these tools to test gene function that are surfacing from our comparative genomics and comparative transcriptomic studies. And so with that, I'm just going to conclude with some of the significance of this work, and maybe I should have started with this, but I think that probably most people are aware that the world is undergoing a global aging phenomena. It's predicted that it won't be too long before the number of old people in the population outnumber the number of young people in the population for the first time in human history. And this is going to have a significant impact both socially as well as economically. The United Nations Department of Economic and Social Affairs reports that the largest or the fastest growing segment in the human population are people over 80 years old. And it's predicted that in the next 30 years, there will be a threefold increase in the number of people over 80 years old and an eightfold increase in the number of people over 100 years old. Now, the fact that we grow old is not necessarily the problem. The problem is that we don't remain healthy throughout our lives, and that aging is the single most important risk factor to a number of diseases, including heart attacks, cancer, strokes, and dementias like Alzheimer's disease, in addition to infectious disease, as we've seen most readily with the COVID-19 pandemic, where the mortality rate goes up tremendously in the elderly population. And so the objective of our work, and for most people who study aging, is not to increase the human lifespan, but to try to keep humans healthier throughout our lifespan, which will not only increase the quality of life for humans and the elderly, but will also decrease some of the economic burden that's going to fall on the shrinking younger population to support the healthcare cost of the aging population. So I'll end my talk with this woman, Mrs. Jean-Louise Calment, who holds the longevity record for humans. She lived to a remarkable 122 years. So this is really just to show that we already have a tremendous lifespan, and really the objective here is to try and keep us healthy for most of that lifespan so that most of our older years are spent like this. And so with that, I want to acknowledge those who have participated in the work that I presented today. We have a fantastic research team at GMGI. We now have 13 investigators on the research team. And so I want to give a shout out to those who have particularly contributed to what I presented today. Jennifer Polinsky is a senior research associate who did the work related to the red sea urchin gene expression studies, as well as the red sea urchin genome sequencing, assembly, and analysis. Kate Castellano is a postdoctoral scientist in the lab. Kate also contributed to the analysis of the red sea urchin genome with those beautiful chromosome alignments in the positive selection analysis, as well as been intricately involved in the cell culture studies. Rhianna McCaddy is a laboratory technician and also our lab manager. Rhianna is a graduate of our Gloucester Biotechnology Academy and a fantastic addition to the research team, and she spends a lot of her time growing sea urchin cells for the NSF EDGE project. And the latest addition to our team is Riz Kellogg. Riz joined us just in December, and their function is really looking at cancer resistance in sea urchins, which will be a topic for a future talk, I hope. We have great collaborators, Greg Ray's lab at Duke University, Gary Wessel at Brown University, and Steve Watts at the University of Alabama. And I really want to give a shout out to our supporters. We couldn't do this work without the support that you give us. I especially want to thank New England Biolabs and Cell Signaling Technologies, who support my position at GMGI in honor of Donald G. Combe, the founder of NEB. The support from the Dusky Foundation at the Essex County Community Foundation allowed us to sequence the genome of the red sea urchin. The National Science Foundation is allowing us to develop those functional genomic tools. And the work we did that I very quickly went over at the beginning on the hallmarks of aging was supported by the National Institute on Aging and a Bermuda Charitable Trust. And so with that, I'd be happy to take any questions that you may have. Yes.
If you could take a red sea urchin and be at the far end of its natural life in what would be normal for that sea urchin, given its ability to take care of business, what would that sea urchin die of?
That's an excellent question. So just to repeat that so everyone can hear, if the red sea urchin lives for this extraordinary life, what does it ultimately die of? And that was one of the very first questions I asked of the fisheries managers, because this is a commercially fished species. It's one of the prized fish species. And if you enjoy sushi and you ever have uni, it most likely is red sea urchin that you're eating. And so when I asked this question, they said that these animals always die from what they call extrinsic mortality, which means something kills them. So they can be eaten by a sea otter, they can have an accident, they can succumb to infectious disease. So there are all kinds of ways in which they can meet their end. It's not through a degenerative aging process. And there's a certain amount of mortality in the population every year. It's just that that rate of mortality doesn't increase year on year. So the older animals have just as high a probability to get through one more year as the young animals. And so they meet their end. Exactly. Yes?
When you mentioned mayflies, which live somewhere between 1 day and 5 minutes, isn't this a bit misleading? Because you really should count its lifespan from when the larva hatches from the egg. It's actually more like several months.
Yes, so if you include its full life cycle, it's several months to a year if you count the larval stages. So that's why I particularly said this is only limited to the adult stage of its life, this short lifespan. Mm-hmm.
What is the difference of the reproductive cycle of the sea urchin that is young versus old?
That's an excellent question. So the question is, what's the difference in the reproductive cycle of the short-lived versus the long-lived sea urchins? And they do have very different reproductive strategies, which is probably essential at driving their lifespans. And so I'd like to equate it to the short-lived species as one of these live fast, die young kind of creatures, very successful at reproducing, produce a lot of offspring, and very quickly develop into juvenile animals. Whereas the long-lived sea urchin spends a lot more time in a larval phase. It has variable recruitment through to adult stages, so not so successful in reproduction, but it goes through many more cycles. So again, they have very different strategies in their reproductive sort of cycles. Yes?
I'm not sure if I heard you correctly. I just wanted to clarify because it's so cool. Did you say that so the radial nerve expression genes are actually increased in the longer that organism stays alive? That's incredible.
Yeah, it was really remarkable to see. The pattern of gene expression was essentially the opposite of what we see in the aging human nervous system. I've never seen anything like it before, and it took me a really long time to come to publishing that work because I doubted it so much and did so many checks to verify that was in fact real. But it does appear that it has a very favorable change in the way genes are expressed in its nervous system with age. Yes?
When you dream about how all of this research would translate into therapeutics that would improve our quality of life, what are the kinds of things that you imagine?
Yeah, that's a great question. And again, I think it's understanding how the processes or the pathways underlying this amazing life histories work, and to be able to find ideally some kind of a small molecule that would improve the function of that pathway in humans. So that, I think, is the ideal scenario. And indeed, we're starting to see some of that when we look at some of the results coming out of short-lived model animals, when you can treat them with certain therapeutics that will extend their life, like rapamycin, for example. And there are other drugs that are known that can extend lifespan. And so it's finding those drugs that are at the intersections of a pathway that has a very wide-ranging effect on biology that I think is going to be key. It's certainly not going to be a one gene here and there type of approach. It has to be something, because aging is such a multifactorial process, has to be something that affects a lot of different systems. Yes?
Has there been any studies done as opposed to pollution and how that affects the lifespan, the factor in there?
Yeah, again, a really interesting question, because and the question is if pollution is affecting their lifespan, correct? Because these are benthic animals, they're crawling around in the bottom of the ocean. That's where a lot of pollutants collect in the sediments, et cetera. That's really interesting, because in some marine animals, we do see when they live in very polluted environments, we see an increased incidence of cancerous types of diseases, for example, shortened lifespan. It really does affect their health. And of course, aging is a combination not only of genetics, but also environment. But so far, I haven't really done those kind of studies to know what that effect is for sea urchins and to know whether the effect of the environmental factors on these animals. But it's a really interesting part of the equation and something that we have to consider as part of aging. Okay. Oh, sorry, one more question. Oh, excellent question, and I always forget to mention that. So there are a number of ways to age a sea urchin, and I depend on my friends in the fisheries in order to get that information. And so one of the ways in which you can age an animal, if an animal has a hard shell and it grows seasonally, it will lay down shell in a seasonal pattern, and you can count growth rings on the shell, similar to counting growth rings on a tree. So that's one of the methods that they use. The problem with that method, especially for the red sea urchin, as this animal is getting up to 200 years, its growth does slow down and the growth rings become very close together, so they're very, very hard to count. And so there are other ways to do it, and the ways that the fishery usually does this is through tag and recapture studies. So they'll tag animals, collect all the animals in a population, generally inject them with a dye, tetracycline, which is incorporated into their shell. They go back a year later, harvest the animals, and look at how much growth has occurred in that 1 year period, and then they can model the growth of the population and extrapolate lifespan from that growth data. Some of the most convincing evidence for the red sea urchin of its long lifespan actually comes from atomic testing, and in the 1950s, where we introduced a lot of radiation into the atmospheres and into our oceans. You can take the red sea urchin skeleton and mill back the shell and count the amount of C14 in the shell, and you will find a point where that signal drops right off, and you know that that is pre-1950 growth. And so I think combining all of these tools together does give us an indication of relative lifespan of these animals. I'm a little bit cautious about these absolute numbers because I think each of these methods have a lot of error in them. So saying something lives to 200, it could be 250, it could be 150. It is a long lifespan, but I don't know about the absolute numbers. But again, the fisheries scientists contribute a lot to understanding the life history of these animals and age them. All right, thank you all very much.